Hi, and welcome again to another episode of Biology Talk. This is Cajigal, and for today, we shall now study PCR, also known as Polymerase Chain Reaction. Siguro naririnig po natin to sa balita o sa radyo itong katagang PCR na ginagamit sa pagtetest sa COVID-19, di ba? So, siguro para malaman ng publiko, ano ba talaga tong PCR na to? Okay? Alright, so PCR is the acronym for Polymerase Chain Reaction. Ito po ay isang common lab technique. Okay, ang ginagawa po nito, itong PCR, is to make many copies of a particular region of DNA. Okay, so you know what DNA stands for. DNA is deoxyribonucleic acid. Okay, so yun ang ginagawa po ng PCR. So pinapadami niya ang isang part kasi ang DNA, it's a long molecule. Okay, so ang ginagawa ng PCR is to make many copies of that particular region of DNA. So, kung baga, ina-amplify niya yung particular region of DNA na yan. So, in a way, parang ano ito, ang PCR parang amplifier po yan ng gitara. Okay? Ng electric guitar. Kasi diba pag twinang nyo, in-strum nyo yung electric guitar, mahina po yan, diba? So, ang ginagawa ng amplifier, pinapalakas niya po yung tunog ng electric guitar. Parang rinig na rinig natin talaga yung mga tono ng strings. Diba ho? So, PCR also does the same thing. The PCR technique makes many copies of that particular region of DNA. Okay? So, parang DNA amplifier po siya. Ganon po ang PCR. And yung DNA region na yan can be anything. Kung anong gusto ng molecular biologist. Okay, so for example, it might be a gene, yung segment ng DNA na yan. It can be a gene whose function a researcher wants to understand. Or it can be a genetic marker used by forensic scientists to match a crime scene DNA with suspects. So, ang PCR po ginagamit na ho yan sa forensic science. So, pag-identify ng mga DNA ng criminal saka ng victim, mga ganyan po. So, ginagamit na ho yan sa NBI. Or even the identification of a viral genome for diagnostics sa medicine. Kaya yun ang ginagawa po nila ngayon sa DOH. So, PCR is used for the identification of the COVID-19 viral genome for diagnostics para ma-identify po natin kung sino po yung COVID positive or kung sino yung COVID negative. Okay? So, yun po ang simpleng explanation po ng PCR. Ano po yung goal ng PCR? So, again, the goal of PCR is to make enough copies of the target DNA region. Okay, so that it can be analyzed or used in some other way. Kasi ang DNA po kasi maliit po yan. It's a really small molecule. So, ang ginagawa ng PCR is to make enough copies of that, in-amplify niya para magamit or ma-analyze in some other technique. So, for example, DNA is amplified by PCR for visualizing by gel electrophoresis. That's another technique po. So, mamaya pag-uusapan natin yung tinatawag na gel electrophoresis. Okay? So, PCR is just one step. Okay? So, in-amplify pa yung DNA. Then, yung resulta niyan, i-visualize by using another technique called gel electrophoresis. So, mamaya pag-uusapan natin yung gel electrophoresis. Okay, uses ng PCR. PCR is used in many areas of biology and medicine, including molecular biology research. PCR is used in medical diagnostics and even in ecology. Yan, ginagamit yan sa pag-identify ng mga extinct species of plants or animals. Okay, so ang PCR po is just to amplify a particular sequence of DNA. Isang step lang po yan. Then, after the DNA has been amplified or we are able to make enough copies of the target DNA sequence, then, isa-subject pa po yan sa isa pang technique called gel electrophoresis. So, we visualize the results of PCR, polymerase chain reaction, using gel electrophoresis. Okay? So, yun po. The results of the PCR reactions are usually visualized or made visible using a technique called gel electrophoresis. So, ano nga ba tong gel electrophoresis na ito? So, this is a technique in which fragments of DNA are pulled through a gel matrix by an electric current. So, ano ba itong mga fragments of DNA? Yung mga, di ba, DNA has nitrogenous bases, adenine, thymine, cytosine, and guanine. Okay? So, these fragments are being pulled through a gel matrix, okay, by an electric current. Kasi di ba yung electricity may positive and negative. So, positive is attracted to negative. So, napupul po yung mga fragments of DNA na yan. Okay, and as the DNA fragments are being pulled by the electric current through that gel matrix, it eventually separates the fragments according to size. Okay? Ayan. So, makikita po natin yan. If you will take a look at this picture, makikita po natin yan as band on the gel. Yung band yung pa mga guhit-guhit po sa gel, which can be seen by the eye. Okay? Again, because DNA is microscopic, maliit po ang DNA, kailangan po natin ng maraming kopya niyan. Lots of copies must be present before we can see it by the naked eye. Okay? Kaya po ang PCR is a very important tool. So, para we can make copies of that DNA sequence para makita po natin, ma-undergo natin sa gel electrophoresis. So, yun ang importante po ng PCR. Yun ang halaga ng PCR. Okay? Yun po. Alright. So, again, saan ba ginagamit itong PCR? PCR is used in many research labs. And it is also a part in forensic science, genetic testing, diagnostics. Okay? PCR is used to amplify genes associated with genetic disorders. Yan, from the DNA of patients. Usually, kinukuha yan dun sa fetal DNA. Okay, in the case of prenatal testing. Okay, PCR can also be used to test for a bacterium or even DNA virus in the patient's body. Okay, if the pathogen is present, it may be possible to amplify the regions of its DNA from a blood or tissue sample. Kaya nga, di ba, ang ginagawa po ngayon sa pagtetest ng COVID-19, sinaswab po yung pasyente. Di ba? So, ang ginagawa nila, kumukuha sila ng sample ng DNA sa pasyente, and then, yung genome ng COVID-19, i-amplify na yan through PCR. Okay, then pag na-amplify na yung viral genome, they will subject it to gel electrophoresis para ma-identify po natin yung kung yung pasyente ay positive or negative sa COVID-19. Okay? So, that's PCR po, simplified. Actually, very complicated tong PCR. Pero ang goal ko po is just to explain it in the simplest way that I can para maintindihan po ng publiko kung ano ba talaga itong ginagawa sa PCR. Okay, so I hope I was able to share some information about PCR. And thank you for listening. Again, stay safe and see you later.